Pathley, for the kind of introduction. Thank you, CPAT, for having us here. So we are the last entrance into the trace world. Our project started about 3 months ago. So as Pathley mentioned, we are in the trace MPS world for pregnancy and women's health. I'd like to acknowledge my co-PIs and collaborators on this project. So just give you a brief overview of the background, why we are doing this microbiological system-based approach for studying pregnancy-related complications and how we can use these platforms for drug development tools. So as you all know that although we wanted to have a happy and healthy pregnancy, most of them will not be happy and healthy, have a happy and healthy ending because of complications of prematurity, which is defined by WHO as less than 37 weeks of gestation. Now, because of this, there are about 15 million cases of premature birth happens around the world, and 1 million of those babies will not survive to see their first birthday because of complications of prematurity. Although many of them survive, they will have developmental diseases and early onset, adult onset diseases in their childhood, like several forms of cancers, asthma, cerebral palsy, many other autism spectrum of diseases. All these are complications. So why can we reduce the rate of premature birth? The challenges are, if you look at pregnancy, there are two individuals in one body. That is the mother and the fetus. And there is no way to recreate the system in vitro unless you take one organ at a time and study one cell at a time. That is what we have traditionally been doing. No animal models, unfortunately, mimic human pregnancy. So there is no way we can recreate human conditions and human pregnancy-related complications in an animal model. So we have to have a humanized system to replicate these data and replicate these pathological conditions and test some drugs. In my experience, there is no new drugs have been tested for the past 30 years to treat prematurity. So there are rising rates of neonatal and maternal mortality has been at the forefront of discussion lately, and pregnant women and children and their fetuses are considered as therapeutic orphans because there are no clinical trials that will enroll pregnant women. Now, there are 5 major organs that actually we need to study if you really wanted to study pregnancy and pregnancy-related complications. One of the major challenges that we have is the feto-maternal interface. That is the organ that we are interested in studying because we're not studying embryonic development. We're not studying fetal organ development. We're studying the interface between the mother and the fetus. That region where the placenta or the fetal membrane barriers actually allows the drugs to cross through that barrier system and reach the fetus to treat the fetus. The problem is we don't have a model to study which drug can cross, what happens during that crossing stages. If they cross, what happens to that drug, what happens to the fetus. So all these things can only be studied using a humanized system. So we, as Pathley mentioned, we have developed 5 different MPS models to study the feto-maternal interface. As you can see here, there are 2 major interfaces. One is based on the placenta. One is the placental maternal interface. The other one is fetal membrane maternal interface. That's what is depicted in this cartoon here. And we first developed a very small 2-chamber chip with minimal complexity, with 1 maternal cell, 1 fetal cells attached to a microchannels. Then we have expanded this to study the entire feto-maternal interface region using 4 different models. And the last one that we can see on this one is this particular one is a multi-organ system where we have combined the entire feto-maternal interface. This whole region is now combined to form this multi-organ chip. This last one is exclusively a maternal lower uterine segment. Again, this is also never studied, so that we have developed these organ-on-chip multi-MPS platforms already, and we have validated this. We have tested this using for multiple mechanistic models, as well as some drug trials. And now this is what we have proposed as our MPS, Trace MPS platform. So each year we will be testing one MPS model, and we will be testing 4 different drugs, and I'll come to the drugs in a minute. So we started this project this year, as I mentioned, and this started with these 2 small 2-chamber chip. This is the choreo-decidual interface, and this is the tissue that we are recreating on this chip. So this is the maternal fetal interface. We wanted to know whether the drug can cross through this barrier. If they cross this barrier, what is the kinetics of crossing? What is the toxicology of that drug? Is there any beneficial effects of that drug on the maternal side as well as on the fetal side? So using this chip, we can actually model that there are 2 cells. One side is the maternal cell, the other side is the fetal cells. These microchannels connect them. And this particular region of the mother also has 30% resident immune cells. We can actually model that also on this chip. So we can actually track the maternal immune cells crossing towards the fetus and causing problems to the fetus. And actually you can test the communication between the mother and the fetus using this 2-chamber chip. So that's exactly what we wanted to study. We have developed the cells for the studies from the both maternal and fetal organs during pregnancy. And we have also developed fetal sex specific, because there is a fetal sex as male versus female has considered as a biological variable, because male fetuses seems to have certain pregnancy complications are much more dominant in male fetuses compared to the female fetuses. So we have developed male and female fetal-derived cells. We also wanted to look at racial disparity, because African Americans have a very high rate of preterm birth in the United States, almost 17%, 1 in 5 almost. In African Americans, regardless of socioeconomic factors, you can see premature birth. So we have also developed race-specific cell lines from intrauterine pregnancy. So that these are all we have for our testing. And the drugs, as I mentioned, we're going to test 2 anti-inflammatory drugs, basically 3 small molecules. 2 of them are anti-inflammatory. One is an antiviral drug. And one of them is a small biological drug, which is exosomal-based interleukin-10 that we have developed in-house in our own lab and tested already that it is efficacious in reducing the rate of preterm birth without any adverse effects to the mother or the fetus. So these are the drugs that we will be testing using these chip platforms. So we will be testing 4 drugs per chip per year. And that's what our model is. And we will be testing these 4 drugs in the next 5 years and using these 5 chips that I showed you. And these are some of the indications that we will potentially can be applied. So what is the context of use for this chip, these drugs? So our goal is to characterize human relevant pharmacokinetic profile of a drug during pregnancy, including the information on drug amounts in different intrauterine tissue. We wanted to see what happens on the maternal side. We wanted to see whether it can cross, and we also wanted to see what is the effect on the fetal side. What happens on the maternal side on the metabolic aspects, and what happens on the fetal side. So as you all know, placenta and the fetal membranes are a highly metabolically active tissue. So that it's not the liver. See, actually, if you look at the enzymes in the liver, you can see exact same mimicry in the placenta as well. So that's what we wanted to recreate using this chip. And the context of use, number 2, is to evaluate the potential of human relevant toxic effect of a drug in intrauterine tissue. So these are the COUs we will be testing. So in our model, we have Dr. Aram Han's lab at Texas A&M University is one of the resource core. Will be manufacturing all the chips. And my lab will be doing all the biological testing. We have the cells. We will be doing all the testing and developing all the protocols. And this will go to Dr. Rosen's lab at Texas A&M University with the standard operating procedures. And his lab will be validating our protocols, our chips, our cells, our methodology, and try to replicate what we have done. And this will eventually go to as the MPS commercialization and technology transfer to the end users. So what have we done? We have started this work, as I said, about 3 months ago. Our administrative core has also already been set up. We have monthly meeting. We have monthly meeting with NCATS, and the FDA representative often present there. We have our own PIs meeting, and we have multiple other collaborative meetings as well. The resource core has prepared and QC'd all the chips that we needed for this year's use. Like I said, I mean, the 2-chamber chip is the one that we are testing, that shown here is the one that we are testing here. We have already started and completed the 3 small molecule trial is already almost completed at the resource core lab, and this SOP will be now moving to Dr. Rusin's lab pretty soon. The qualification core, Dr. Rusin's lab, will be testing all these SOPs that we will be providing them, and they are also in the process of validating multiple platforms, validating the assays. They are in the process of developing some of these assays and testing some of the other parameters that they wanted to use for qualification purpose. So with that, I just wanted to introduce our team. So these are the administrative core, resource core, and qualification core. We also have a specialist, maternal fetal medicine specialist on our panel to give us some advice on the translational side of these things. That I want to thank CPAT for the opportunity to come and give us this opportunity to talk about our Trace Center. Thank you.